Good morning, everyone. Well, thanks everyone for being here again in our PGBQ Symposium, our second edition. And I thank Dr. Lane, and she's here with us, honoring with her presence. And I'm going to ask my dear friend Elba to introduce Dr. Lane's lecture. So Elba, please.
Okay, good morning, everyone. Good morning, Dr. Lange.
Good morning.
Good morning, Anderson and Luciana. Congratulations for the organization of this meeting of our biochemistry graduate program. We have trouble learning the remote mode of everything, but we are moving forward. And well, it's a time of change. And after the COP26, I expect that activities such as that Dr. Lange work with and has been working for many years will be even more relevant. And so Dr. Lene Lange is the Director of Bioeconomy Research and the Advisor of Denmark from the Technical University of Denmark that I have myself some times ago the opportunity to visit. So it's an honor and it's a pleasure to have here you and your availability, isn't it? Okay. Professor Lange has been working mostly in the field of applied microbiology and molecular biology, biomass, and microbial enzymes. Is there anything more important than that? I don't think so. For these times of the mankind history. Her prolific career includes 282 scientific publications, it's a lot, with over 29,000 readings. And in her studies, she has emphasized enzymes and fungal biotechnology. Today, Dr. Lange will give us a talk on functional annotation of enzymes that will span our learning in terms of enzymes possibilities and evolution of fungal secretome, which is also a very important tool for us to discover new enzymes. And thank you again for being here. And please let me know when you are ready to start your presentation, Dr. Lange.
I'm ready now. Thank you.
Let's move forward.
Would be, thank you for the nice words and thank you for inviting me, giving me this opportunity. If I can have my slides, I will just be starting. Time is precious and I have a lot on my mind to say.
Oh, wonderful.
There you go.
Thank you so much. My text here, the functional annotation of enzymes, gives insight in evolution of fungal secretome. And remember, the fungi, invasive power of fungi comes from what they secrete out of their hyphae. That's biology. Next slide. And just to put it in applied perspective, efficient and responsible use of biological resources is a necessity. They do not talk enough about this in Glasgow in COP26. Action is needed. We waste way too much, but it's at the same time a huge opportunity. So much waste can be created value from. I'm not going to go through, but yeah, you can have my presentation and you can see all what we are wasting. 30–40% just of food waste lost globally. And then for all the side streams and the type of residues, we do not upgrade with enzymes. It's almost half of all what we produce, which is not used properly. So to make use of all that beautiful resources, we need to find new enzymes, more optimized and more efficient. And remember, feed higher value product, but also feed takes up 75% of all arable land in the world. So if we can produce animal feed of high quality from what is now wasted, we can make space for biodiversity as well. I have given references to the different points, what I'm going through, and they are at the bottom of the slide. Next. Just to remind you, using side streams and residues and waste is not just energy. Energy is the lowest value at the bottom of the value pyramid. Energy and heat is low value. Bulk chemicals rather low. Fuels very also low. You have to go all the way to food, feed, health promoting to get the highest value out of it, and both for food and feed. And next. And then my point here, in order to be really using the strongest card we have to go for the time and cost efficient discovery of the best enzymes, then it is not enough to do what most have been done, which is alignment-based annotation. Because alignment-based give equal emphasis to all sequence, all parts of the sequence. What we know that evolutionary pressure is conserving and being taking more important to certain parts of the genes. And therefore we take advantage of that alternative, non-alignment based functional annotation. The first version was called peptide pattern recognition. The invention were twofold when we developed this in 2009 to 13. The first step, but if you take a group of protein, all proteins in the world, you can group them according to sharing the having the same type of conserved peptides. Imagine that different enzymes have a kind of signature patterns. We can recognize those, and the ones which are sharing similar signatures belongs to the same group. That we can cluster it into a group, and that is the PBR clustering, peptide-based clustering. Invention 2 is, and that was my inventive step among Peter Busk and myself. Peter Busk did the bioinformatics, found the way of clustering, and I had the divine inspiration and said, maybe the signature peptide, the unique conserved peptides, maybe they are actually the key for recognizing the function. So for that, next slide. Just to say short, here is a presentation. If you go back, this is what I have been doing in private sector and public sector, advisory and research, etc. But I want to emphasize in my long career, both in private and in upper management position in academia and industry, I all through my career had my own research group. That's why I have many publications, even though I have high level management position. I don't think you can be a really good leader in R&D if you also know what is the frontier of your field. So next slide. Next, please. Could you go to the next slide, please? It's not working. Could you move to next? Thank you. So after the peptide pattern, we found out there were some problems with the PBR. And we made a new one, we call it conserve unique peptide patterns. For here, we actually managed to improve it in a number of different, and that's too technical. But I just want to say we ended up with a conserved unique peptide patterns, which has improved sensitivity and precision in prediction of function. It was simply a better, more precise and more sensitive method. So this is the one I will focus on. Next one. Here is the stages, and this is bioinformatics. And I simply want for you, the ones who are not bioinformaticists, among us, most of you aren't, I know. But then remember, first was clustering, the ones who share the same peptides. The next was predicting what type of function is the signature peptide typical of. Next slide. So then the next part, for you who know, we need to understand what kind of annotation is telling us. So we have families, protein families. We have also in enzymes, the carbohydrate active enzymes, different types of proteins. Then we also have subfamilies, but still with the subfamilies, there are several different functions in each family. So the point with the cup is that when you have a clustering by cup, you have the same function to all the proteins clustering in the same group. And that is, again, therefore, you can predict if the function of a protein, if just one member of your cup group has been characterized. That is efficient use of genome sequences and the very sparse characterization. Too few enzymes have been characterized. Next slide. Oh, it takes a long time to change. I need to use my time better. Okay. Here, this is too complicated to look at, but I just want you to dream and look at this one. Dream. The outer circle is actually a dendrogram of all enzymes belonging to the enzyme, the carbohydrate active enzyme, family number G830, glycohydrolase 30. And then what it can be shown in the inner circle, you can see there are different colors, and those colors are the subfamilies of the proteins. Next slide. In the next slide, I will show it a bit in a bit larger, where you can see a little bit more. The red is the subfamily 5, and that includes 4 different subgroups. That means that we now know not just to annotate to the subfamily, but we know which cup group it belongs to, and therefore we know the function. So now we can start biology when we know the function. This is a real movement, and from data and a lot of genome sequence data, we move into understanding function, meaning we can start to talk biology and function in use. Next slide. I would like it here, and that is also very busy, but don't give details. Just want to say, cup can be used for all types of organisms. Here, it is a case where we analyze a protozoa, an animal, labyrintula, and find all these different types of enzymes, even predicted to function. So that means that the cup method available as a web-based platform and online platform for automation can be used also for annotating plant enzymes. Next slide. Applied cup application, just to mention, we can demonstrate gut health effect of ingredients by actually finding changes in the microbiome composition and changes in the digestive function profile. So not just one organism at a time, but even a microbiome can be analyzed, and we can find out feed ingredients. For instance, I know we can make a feed ingredient in Brazil. We worked on that from the peels after the press pulp, after you have made an orange juice. There are short oligos of the fibers can have a beneficial effect on the microbiome of the gut health, improving on both animal and people. All that can be characterized by changes in enzyme profile, which is possible by cup. Next slide. While you're changing, I want to say they just started a construction work in my street just outside my window. I hope that is a noise behind, and I cannot do anything about it. I hope you can still hear me. So if you can hear me, that's good. So functional annotation alone is, however, not enough to understand evolution of the fungi or other types of species. The key is, in order to understand evolution, you need to integrate the 2 features of the enzyme. Most publication, if you go through it, either describe the family of a protein or the function at 2 different lists. What I'm saying is that for each of the sequences, each of the enzyme gene, you need to find out what protein it codes for and what is the function. It is an integrated one observation We call it the function family observation, FF observation. And this is try to understand evolution. Evolution is putting pressure on new species to be made. I have to leave off. So in order to be the one surviving evolutionary pressure, you need to have the optimal digestive function in the optimal type of protein. So both function and protein should be optimized. And that to understand that is that different type of protein structures, independently of its function, is actually determining how close it can get to the substrate, size and shape, steric shape, but also the stability of the enzyme, the pH and temperature tolerance, and optimum. So the protein is important for the fitness. So both the function and the type of protein should be the right in order to be the one preferred. So creating the best fitness of the species. So that means that digestive enzyme secretome is an integrated part of the evolution of species. Next slide. So using this observations, then if we take all the fungi ever sequenced, that's almost 2,000 genome sequences with good quality genome sequences, then we can make this enzyme profile relatedness analysis by using this function family integrated observation. And can you see, on top is the Ascomycota, the one group of fungi, and the, below is the Basidiomycota. And the whole figure is representing the different position of all the sequenced species. As you can see, it clearly separates Ascomycetes from Basidiomycetes, top and yellow and reddish, but it also distinguishes the clustering of Aspergillus and Penicillium to the right of the red one. Next slide. Then we go one step further. We made the hypothesis. What if the hypothesis, phylogenetic relationship of an organism, which we normally see in a phylogenetic tree. If we made that based on the enzyme profile relatedness, meaning function family observation, if you have them or not have them, then could that, if it is just as good in a phylogenetic marker, the trees would be identical in synchronized matching. We chose a complicated, two big, huge genera, Aspergillus and Penicillium, and then we tried to go through your Charcot calculation, comparing what, making the relatedness, stronger relatedness if they have the same type of sharing many of the same observations. And we hope that it would be made two trees which were identical, but it didn't. It did not confirm our hypothesis. And then I got this idea, and that was, I was in Costa Rica along with Jens Friesfeld, in my college friends from DTU, and me, a colleague, very good colleague, and we got the idea. Maybe Euler calculation only counting the ones they share having. Maybe if we also counted the what they shared not having. Can you see what they lost? Is also a part of evolution. And then there were a total congruent figure. So here, I'll show you next slide, then you can see it. And this is so beautiful. But just look at the sun as a beauty. You cannot see all details. But here, the calculated Kazan function family profile is made into a diagram and a dendrogram, and that is the tree in the middle. The little tree in the middle has a total congruent with the section of all species, section of all grouping of species based on the normal phylogenetic markers. So the outermost ring shows all the sections of Aspergillus and Penicillium, and they have exactly the same. So here, conclusion, we can use, this is an integrated digestive enzyme secretome composition, is an integrated part of speciation. It gives fitness, and it is therefore, as you can see, it is congruent with a organismal, normal taxonomic grouping. And that is quite revolutionary. Suddenly we come and say that the enzyme genes is part of genotype through a not just part of the phenotype. And that is the phenotype is a normal enzyme is normally seen as a phenotype. So this is a real evolutionary, highly remarkable observation. Next slide. Now we want to use that even more, because we went from COP, the finding a way of actually functionally annotate. We found out that we could make these function family observations and make enzyme profile relatedness across all fungi sequenced. Then I got the idea. What happened if we try to sum up all the function family observation in a genome sequence fungus? Because if you sum up and look at the ones who are having the highest sum, the ones having the highest sum of function family observation has the richest function specificity diversity if you only count the unique function family. A unique function family is a protein type of protein with a specific function. So the one who have in the highest number of such function, family function observation, different observation, is the richest Kazan function specificity. So that fungus you find with that is the top scoring with it's a gold mine for finding new enzymes. But another kind of top scoring is if you add both the ones, all the unique ones, but also the redundant ones. So if there are several copies, you count all the copies as well. There, by this one, you find the fungi which have the highest total degrading capacity. And can you see here? I told you about how you can use it to find the best enzyme function, but I also here tell you how you can actually use this function family observations to find the strongest. For instance, if you have a big collection of your available strains you are interested to work on, you can immediately find out which one is the richest in diversity and in total degrading capacity. So we can find, and this is also published, the references below, and that's from 2021. And you will remark, I hope you'll see all of my references are for the very last years. I've never been so productive in my life in science before. So we can find the richest species in cellulolytic, breaking down cellulose, breaking down silanase, breaking down pectin, breaking down lignin. All of that can be done in this, and you can look up and find the species closest to the one you have and be inspired which one to look at. So also the interesting part, it's not like you can say the strongest enzyme will be found in this and that type or that ecological or that habitat or something. It's found, the best one are found in many different places. Even a rock degrading fungi, inhabiting stones with only very little organic material ending on the stone. And why is that so high? It needs to be so good to degrade whatever lands on that stone. So it is so important for its survival and fitness that it has all available enzymes. So next slide. And this is a very technical one, and I'll not go in for the one who are really interested. This is a method for hotspot analysis in a graphic abstract, and where you are going from one to the other and ending up with finding the hotspots. So let us immediately go to the next slide. And here is one of the surprises. We got many surprises. With the rock fungus was one. Another example of surprise was in some of the hotspots was actually endophytes, fungi, which are growing inside a plant endophytic fungi are also rich in enzymes. I would just wonder, because they are not destroying the host, but still they need to go through many types of tissues and structures, so they need many. But the one which is actually important also in Brazil, I wanted to show with you, and that I think it was the genome sequence was actually, I think, deposited from Brazil, if I remember correctly. And that is the top scoring here, Mycena citricula. And that is a coffee pathogen, and it's a basidiomycete. And normally we see ascomycetes to be stronger in enzymes than basidiomycetes. But here, in pectin degrading, we found a Mycena citricula, a small cap fungus, to be, you can see the second column called pectin. It has the highest 204 observations of different pectin degrading observations. Next slide. I want to show you a small picture of Mycena citricula. Here you can see it on a coffee fruit, and if you look at the leaves of the coffee plants, they are actually making holes in the leaves. The fungus is so efficient in breaking down the plant substrate that it makes there are whole holes which disappears. And you can also look at the berry of the coffee. It has a flattened surface where the fungus is growing. That it is destroying all of the tissue underneath, so it kind of sink in. So the rounded shape is lost. So because it is digested by invasive power of this fungus. Next slide. Impressive fungus, very little, very tiny, but very impressive. Then I want to just share with you a few amazing things. I have done, I'm one of the few surviving experts. I mean, I learned from Professor Sparrow way back about the ancient fungi, and most people left the field. Not very, very few people work on ancient fungi. And here is one example of the beauty of a very, very simple enzyme type of fungus. It is just one round cell with some rhizoids. And in the low picture and the upper picture, you can see it is growing in eating the filter paper fibers. And the one I started was Rhizophyctus rosea as a model. And if you look at a phylogenetic tree here on the type of cellulose degrading enzyme called AA9, you can see there are many such enzymes. The green ones are the types found in ascomycetes. The blue ones are found in basidiomycetes. But what happens is that you can see the red ones. And the red ones are all the ones which are actually found in Rhizophyctus rosea. And that is amazing that you have so many already in the ancient fungi had such a rich diversity of enzymes. So that is, I think, amazing observation, and I hope you enjoy looking at that. I think it's amazing. The next part, next slide, if you look at that. Here is another interesting discovery. Enzyme discovery for degrading keratin by peptide-based. So we make the discovery by peptide-based functional annotation, and we try to find new, elucidating how can fungal enzyme degrade the recalcitrant keratin, which is found in like chicken feather, hair, many places. And based on our discoveries using COP and PPR, we are finding the following, or made up the following hypothesis. The proteinaceous structure of keratin. Remember, keratin is protein. It's not like cellulose or something. It's protein. The proteinaceous structure of keratin can be decomposed by a synergistic effect of 3 types of proteases. An endoproteases, that's the red one. An exoproteases at the end of the helixes, and that's a green one. And oligopeptides, and that is the orange ones, shown to the right with cutting the small oligos. And if you feel that you have heard about that kind of story before, I can tell you that's because that's exactly the same as when you cut down chitin. There are the ones in the middle, the endoacting, there are the exoacting, and there are the ones functioning on oligos. So it's similar, even though that is an enzymatic strategy and goes on polymers made of cellulose, of chitin, but also here a protein. And then there is also a disulfide reductase, which is cutting in between, which makes the recalcitrancy of keratin is these disulfide bridges. So this amazing structure is possible to elucidate with this complexity, to act to this level based on that we have function. Functional annotation is key to understanding this kind of complexity. And what is this sounds very basic research, but to me it's very highly applied, because by this hypothesis, industrially important, now we know what type of three to four types of enzymes we need to mix in order to have a blend which can make convert all the huge amount of chicken feather which is mounding up in the world without any use. Because if you eat most of the just eat feather directly, it goes through most of the animals without getting any nutrient from it. But here you can expose and it can become a protein-rich animal feed. So in that sense, for me, using functional annotation is a fantastic area, because you can study across the fungi, across all types of organisms. You can study interactions between organisms. Instead of just in genomics, we mostly study one organism at a time. But biology is interaction, but also applied use of enzymes and upgrading of waste is also interaction, that we need secreted enzymes which interact and degrade or modify substrates. So in that sense, there's a very short way in this type of research, and that I think if many of you PhD students and postdoc, it's fantastic to choose this kind, using this kind of tools, because we have now at this wealth of genome sequences, and we can go from being just data, but actually giving information and insight, understanding of biology, but also insight which can make it more easy for us to really make use of all what we are now wasting. So in my final slide, and now the sun went behind a cloud again. So I think I went back to my normal color picture. Sorry for that. I had not expected any sun to come out today, but it came. Maybe just waving. Sun is also in Denmark, not just in Brazil. So maybe it was greeting you. But let me here give my closing remark with the UN Sustainable Development Goals at the corner, remembering that is our framework, our language, our common language, which they should understand fully in Glasgow. And I really hope they will understand it better because all those 17 Sustainable Development Goals will not be achieved in adaptation to climate change mitigation, adaptation to climate change. We need to use the resources better. You can see, for instance, now I'm going a little bit outside my screen. I just want to convey to you, Elba, you started by saying Glasgow, and I say we have to remember that there are a challenged food security in several areas already in Africa. So this climate change is not just their rising water, it is also resources and food security which is at risk. And there, what is the first we can do, and the foremost easy to do, is not just to be the long haul in making stronger plants which can grow more even though they are challenged by climate change, but the short low-hanging fruits of using upgraded use for food and feed and health promoting product, including gut health and skin health and wound, many health promoting efforts from what we can get out of what is now being wasted. So the closing remark, circular bio-based food from residue, side stream, etc., represents such a huge opportunity for improved food security, for improved nutrition, gut health, from improved climate Climate change mitigation, you need to use the resources better. And we do that by upgrading circular use of our resources for food, but also for cotton and the other material. And then also for jobs and local livelihood. For upgrading of local waste is also creating jobs and development in the places, also in rural areas. And it's globally as well as locally. And for all this to happen, we need both the best enzyme and the optimal microbes. And optimized targeted enzyme discovery, going beyond really using functional annotation, is key for making this vision of improved use of resources for the benefit of higher sustainability for all of us and for a better planet, better for people and planet and environment and biodiversity, better room for all of us. Thank you so much for listening.
Anderson, should I?
Thank you, Professor Lane. It's a very excellent talk. Thank you very much for that. And Professor Elba?
Thank you very much. Dr. Lange, congratulations for your presentation. We need more people working with enzymes all around the world. This is a very interesting tool you presented because simplify everything. So you can have a better look at our resources just using these tools that you have just presented. May I make some remarks, specific remarks, Luciana? Or
I would love to hear what you want to say.
But maybe the presentation is also open to the general public. So I don't know if I'm going to trespass in line here or not.
No, you can't go forward, Elba.
Okay, thank you very much.
Okay.
I have been working with biomass enzymes for a long time now, and I love this subject. It is my area of choice in terms of research. I have been doing this for a long time. And I noticed that you not even mentioned the genus Trichoderma.
Yeah, I want to, that's a very good question or remark. The surprising result of the hotspot analysis is that Trichoderma is not an outstanding enzyme producer. Not at all. The point is that the outstanding part of Trichoderma, where it is outstanding, and that is a damn good secretor of enzymes. And that means that it is, so it's not enough to have the interesting enzymes. It is also important that you are able to, from the tip cell of the hyphae, to secrete so much, so it really makes the biomass conversion. So it's where it excels is in secretion. And I would have to say that almost all of the industrially used enzymes and species, species of fungi, are not among the very high excelling in diversity, function specificity diversity, and neither in total capacity. And that, to me, that tells that there is a lot more to be found.
Okay. And this puts the all enzyme industry in terms of biomass degrading enzymes upside down, isn't it? Because Trichoderma are the main enzymes are the main ones that present. Yeah, well, this is interesting. We have noticed something like this in our research also. And may I ask a very challenging question for you?
It's very welcome.
Bioeconomy. It's that Denmark, likewise Japan, alongside Japan, has been the cradle of enzyme technology. I have visited the research centers, and I remember the early presentations on amylases, transformations, and genetic engineering to get better amylases and so on and so forth. So it's the cradle. Likewise, Novozymes. It's just a natural development of research and needed to use and company development. And I have also visited Novozyme in Denmark, and I have a wonderful memory of the facilities and everything else. This is one side of the story. The other side of the story is the availability of enzymes, industrial enzymes, in the places where they have the researches, which is Brazil, mostly Brazil, because we are huge, we are relatively underpopulated with a lot of space, and we can produce a lot of biomass without degrading the environment. But we don't have enzymes over here. There is a strong policy of the international enzyme companies to not allow in situ enzymes production, to not license their technologies. And this is a huge barrier. And it's almost difficult when one small company starts working with enzymes production, and if it has some value, it's bought. And that's it. End of the story. To me, it's a nonsense.
I think I understand what you said. In order for bioeconomy to really contribute significantly and sufficient to development globally, I believe that it is necessary to actually also have many more local activities.
Yeah.
Many more flowers blooming, yeah? And I have done two things. The one in that respect, one thing is that I hope that the paper on the hotspot paper will inspire you to actually start looking at some of the fungi where you can actually, by growing them, non-pathogenic, good, easy, choose the right ones who are not toxin, mycotoxin producers, etc. Clever choice, and try to grow and make a blend of enzymes from that fungus. And use that without having, you can use that without having to ask anyone, because that is a non-GMO, and you can use the blend naturally producing. And if you look into the hotspot paper, you can find species which are the best for the pectin degrading, best for the lignin degrading, best for the cellulose. So you can also go and find not just the general biomass conversion, but also this one. So that's one point. So I'm trying to empower more people working on enzymes to get the use of commercial use and actually of your research. I have been several times to Brazil, and I know what you are talking about, because you are not really implementing all what I'm talking about in upgrading industrial side stream. Is not, that step is not yet taken in Brazil, at least not to a very large extent.
Not so far, yeah.
Yeah, and that's what I think you should do. So I'm telling we should. Then the other part, I'm the vice chair of the scientific committee of the advising EU Commission in the bioeconomy. And here we are talking about an international strategy where we should be better in actually communicating, and especially I've seen a huge potential in working with the big biomass degrading, biomass producing countries, Brazil, India, China, huge, which have not taken the step of actually developing upgraded use of so much of enzymes, and then upgraded use of your residues and side streams. So there is also in my first slide, there was a reference to this describing what has been done in EU, and that we have gone to the point where we are optimizing in not just fuel and materials, but doing all the ones which is interesting for Brazil, food and feed, higher value products. The last thing, and now I will shouldn't be so long in answering, but the other thing I'm advocating, I'm a member of the advisory board of MIRI, which is all culture collection in Europe. And I want us to make an open access biology package where there will be organisms which I have no patents, no strings attached, free to use and safe for production of enzyme, and also for production of, for instance, or used as a host for heterolog expression, higher yield. So if we, in this way, I want to empower exactly what you asked, the new next wave. And I can tell you, I have been talking about this also when I was research director in molecular biotech in Novozymes. And you know what my bosses say? They didn't say, oh, shame on you because you are destroying our business. No, they said, what you are distributing of knowledge is actually the way that you prepare our future customers. When they have tried to use their own, then they will, if they grow to bigger, then maybe they will buy ours. Yeah? So I'm just saying that I'm not going behind the back of my former bosses, and I love Novozymes. It's a fantastic company. But I just say that it is so good that we all parts, also in Africa, most needed in Africa, use very simple trichodermas, certain parts of non-toxic penicillium can be used. There are aspergilla. There are so much which can be done without infringing patents. The whole base of bioeconomy technologies, which are open access. And all what I have described here is open access. COP is open, PBR is open, free to use.
Yeah.
Okay, we have a few questions from the chat. So everybody's so excited with your presentation, Lene. And Professor Rafael asked, is possible to adapt or train CUP software to other enzyme classes besides sugar degrading enzymes? Is your grouping working on that?
We have focused on enzymes. However, we have also been used for proteases, and we have also been used attempting using for proteases and attempting use for lipases. It's doable, and if you want to, you can go ahead and try it yourself by going through the clustering only and see your result, what will happen. But it is, I think it's for future research to be done, and the proteases, I really want this to be done. But right now, I don't have funding to do it, and I have so many other projects to do. But I think it is next to be done. But other annotation tools are also available for proteases. And try to look into our keratin papers. We are getting close to using it there.
Okay, one more question from Professor Ayla. Fantastic talk. Could you comment a bit on the decision to make CUP an open tool, and if there are difficulties to acquire funding to keep it running for a long term?
But CUP is open. It's not just PPR. CUP is open to use, and it's available in an online web-based version. So It's automated, easy to use. And please go ahead. Too few people find it. And it's a pity, but because most annotation is now done by the big companies, and JGI, they use the PPR hot pep integrated in DBCAM too, but we are not the CUP, and I think they could do much better if they actually included also CUP functional, the power of CUP. So, but it's open. Everything I have worked with is open access. Please use it.
Okay. And the last question is for our student, Priscilla. Dr. Lange, thank you for your talk. It's amazing. What is your opinion about the major challenge in order to reach a more sustainable society by using enzymatic machinery? A philosophical question, right?
No, I think it's very pragmatic. I think it's actually to the point.
Yes.
I think that my dream scenario is that the CUP focus starts to include resources. And if they include resources, they would see the fantastic opportunity in actually upgrading all what we now waste.
Okay.
Going down, climate change mitigation is it's a strong contributor, but also to food security. So I think the people's attention to food security being endangered, and people's attention now, politicians' attention to biodiversity crisis, is both pointed in the right direction, using what we now waste through use of enzyme, upgrade by enzyme. Then we can leave more room for biodiversity, and we can get more food for people and feed for animals.
Okay, thank you very much, Professor Lene. Thank you, Professor Elba. We're going to continue our symposium in another link. Thank you very much.
My pleasure. Thank you very much.
My pleasure. Thank
Okay.
you.
Bye, Lene. Bye, everybody. Thank you.
Bye-bye.
Bye.
Thank you so much.